Would you eat food made by billions of microorganisms? The truth is, many of us already do every day. Good day, everyone. Today we will discuss Laboratory Activity 7, the microbial fermentation in wine, kimchi, and sauerkraut production. Fermentation is a fascinating process in which microorganisms transform simple ingredients into products with enhanced flavor, preservation, and nutritional value. Through this activity, we observe how microbes contribute to food production and the important changes that occur during fermentation. Now, let's proceed and hear it with my group members.
Fermentation is a biological process where microorganisms such as bacteria, yeast, and fungi break down sugars and other carbohydrates under low oxygen or anaerobic conditions. During this process, organic acids, alcohol, and gases are produced. Fermentation has been used for centuries in the production and preservation of food and beverages because it improves flavor, texture, nutritional value, and shelf life while helping maintain food safety.
For the 3 experiments, we used green cabbage, Chinese cabbage, carrots, radish, and ripe mangoes as the main substrates for fermentation. Salt, sugar, chili powder, garlic, ginger, and baker's yeast were also used as ingredients, while 70% alcohol was used for disinfection. The apparatus and equipment included glass fermentation jars and bottles, mixing bowls, a kitchen knife, cutting board, food processor, measuring spoon, fine mesh strainer, and a tablespoon for compressing the vegetables. To monitor fermentation progress, pH strips, a pH meter, labeling materials, and observation log sheets were utilized. A plastic balloon was also used as an improvised airlock for the mango wine fermentation. To maintain aseptic conditions and minimize contamination, personal protective equipment consisting of a laboratory gown, surgical mask, hairnet, and disposable gloves was worn throughout the experiment.
Before conducting the experiments, aseptic technique were strictly observed by the group to minimize contamination and ensure the reliability of the results.
The production of mango wine fermentation began with the preparation of mango juice. 4 fresh ripe mangoes were thoroughly washed under running water to ensure cleanliness. The mango flesh was then carefully removed and placed into a food processor where it was blended to extract the juice. The processed mixture was filtered using a fine mesh strainer to remove large, solid particles, resulting in a smoother mango juice. And 2 cups of sugar were added. The mixture was stirred thoroughly until the sugar was completely dissolved. After preparing the mango juice, the inoculation process was carried out. A pinch of baker's yeast was added to the juice and gently stirred to ensure the yeast cells were evenly distributed. The inoculated juice was then transferred into a clean glass fermentation bottle. Adequate headspace was left in the bottle to allow for gas production during fermentation. To serve as an improvised airlock, a plastic balloon was securely attached to the mouth of the bottle. The bottle was then properly labeled for identification.
On day 0, the mango wine had a yellow color, sweet odor, slightly pulpy texture with visible gas and bubbles and a pH of 5. On day 1, the mango wine maintained its yellow color, developed a sweet and fruity odor, had a clumped and sticky texture with visible gas and bubbles, and a pH of 4. On day 2, the mango wine remained yellow with a sweet fermented odor, clump texture, visible gas and bubbles, and a pH of 4.07. On day 3, the mango wine retained its yellow color and sweet fermented odor, with more liquid visible, continued gas production, and a pH of 4.07. On day 4, the mango wine remained yellow with a sweet fermented odor, clump texture, reduced gas production, and a pH of 4.03. On day 5, the mango wine remained yellow and developed a sweet fermented and alcoholic odor, with sediments forming at the top and a pH of 4.02. On day 6, the mango wine changed to a yellow gold color, developed a stronger alcoholic aroma, showed visible sediments and minimal gas production, and had a pH of 4.01. Day 7, the mango wine maintained its yellow gold color, sweet fermented and alcoholic odor, mostly liquid texture with visible sediments, and a pH of 4.01.
The vegetables were thoroughly washed. After that, it was cut, salted, and left to stand for 1 to 2 hours to draw out excess moisture. It was then lightly rinsed and drained to remove excess water. A seasoning mixture composed of garlic, ginger, and chili, and a small amount of sugar was prepared and added to the vegetables. Finally, all the ingredients were mixed thoroughly using gloved hands and a clean spoon to ensure that the even distribution of the seasoning.
After seasoning the vegetables, they were transferred into a clean jar. The vegetables were pressed firmly to remove trapped air and ensure proper fermentation. They were kept submerged in brine as much as possible, while enough headspace was left at the top of the jar to allow for gas production during fermentation. The jar was then covered loosely or opened daily to release any accumulated gas. Proper labeling was done to identify the sample and fermentation date.
Day 0. Freshly prepared kimchi had a strong spicy smell, firm texture, and visible bubbles. The pH was 5, indicating a slightly acidic condition at the start of fermentation. Day 1. The kimchi remained red with a strong spicy smell while the seasonings became clumpy. The pH decreased to 4 to 5, showing the beginning of acid production. Day 2. Larger bubbles appeared, indicating more active fermentation, while the spicy smell remained strong. The pH reached 4, reflecting increased acidity. Day 3. A fermented aroma began to develop, and the brine partially dried. The pH further decreased to 3 to 4, indicating active lactic acid fermentation. 4. The sour fermented smell became more noticeable than the spicy smell, and the kimchi appeared drier. The pH remained at 3 to 4. Day 5. The fermented smell intensified and only minimal brine remained. The pH stayed at 3 to 4. Day 6. Fermentation continued as the fermented aroma became stronger and larger bubbles persisted. The pH remained 3 to 4. Day 7. The kimchi reached an advanced stage of fermentation, characterized by a strong fermented smell and active bubbles. The pH was still 3 to 4, indicating that the acidic condition was maintained.
The process started with preparing the cabbage, which was thoroughly rinsed with water before discarding the damaged outer leaves. Cabbage was then sliced into thin strips and transferred into a clean bowl. Salt was added and mixed well, followed by a firm massage to break down the plant's cells and draw out the natural juices needed to form the brine. Next, the mixture was transferred into a clean jar and packed tightly. It was pressed down firmly to eliminate trap air pockets and ensure the cabbage remained fully submerged in its own brine, creating an anaerobic environment. Adequate headspace was left at the top of the jar to accommodate expansion before it was covered loosely and labeled for tracking.
In day 0, the sauerkraut had a pale green color, clear brine, fresh odor, watery texture, visible bubbles, and a pH of 5, indicating the start of fermentation. For day 1, the sauerkraut became pale yellow-green, with a soaked yam, bean-like odor, while remaining watery with bubbles and a pH of 5. In day 2, a more prominent yellow color and salty odor were observed, with bubbles present and a pH of 5, showing continued fermentation. In day 3, a brine turned cloudy yellow and developed a more fermented odor, while the pH remained at 5. In day 4, the sauerkraut had a cloudy yellow brine, salty cabbage fermented odor, bubbles, and a pH of 5. In day 5, the fermented odor intensified and the pH decreased to 4 to 5, indicating increased acid production. In day 6, the texture became sludgy, mold appeared, and the pH dropped to 3 to 4, showing advanced fermentation. And lastly, in day 7, a stronger fermented odor, a sludgy texture, a widespread mold growth, and pH of 3 to 4 were observed, indicating spoilage.
For the results, in fruit wine fermentation, the wine retained its yellow color, but its aroma had major shifts from sweet fruity scent to a distinct alcoholic scent. Gas production was also highest during the first 3 days, as shown by the balloon inflation. Its pH also decreased from 5 initial pH to 4.01. Moreover, in kimchi fermentation, it maintained its red kimchi color, while its odor gradually changed to a more sour and fermented. Bubble formation was also present, indicating active fermentation. Its pH also showed increasing acidity of 5 to a range of 3 to 4. Lastly, the sauerkraut fermentation, the color changed from pale green to yellow, with the brine changing from clear to cloudy. In addition, bubbles were also present all throughout the observation period. Its pH also decreased from 5 to a range of 3 to 4.
Presenting the results, let us now explain these observations using microbiological concepts. The microorganisms involved differ across the samples. In mango wine, Saccharomyces cerevisiae yeast drives alcoholic fermentation, producing ethanol and carbon dioxide, while in kimchi and sauerkraut, lactic acid bacteria such as Leuconostoc, Lactobacillus, and Weissella dominate, yielding lactic acid, carbon dioxide, and other metabolites that enhance aroma, flavor, and texture using natural sugars in mango, cabbage, and vegetables. Yeast in wine requires anaerobic settings to maximize alcohol yield, while lactic acid bacteria thrive in oxygen-limited brine environments. Finally, fermentation acts as a preservation method. The combined effects of ethanol, acidity, and microbial competition inhibit spoilage organisms, extending shelf life and ensuring product safety.
Throughout the activity, strict safety and sanitary procedures were implemented. To minimize microbial contamination, personal protective equipment were used during the experiment. The laboratory workspace was disinfected using alcohol before and after the procedure. To ensure aseptic conditions throughout the experiment, all equipment was thoroughly washed, sanitized, and disinfected. The fermented products prepared in this laboratory activity were for observation only. None of it were consumed because it was prepared for academic purposes and not under food-grade production conditions.
Microorganisms played a vital role in the fermentation of mango wine, kimchi, and sauerkraut. During the fermentation process, various physical and chemical changes were observed, including changes in pH, odor, texture, color, and gas production. These changes served as indicators that fermentation was actively taking place. In mango wine, yeast converted the natural sugars present in the fruit into alcohol and carbon dioxide, resulting in the characteristic aroma and flavor of the fermented beverage. In contrast, kimchi and sauerkraut relied mainly on lactic acid bacteria, which produce lactic acid and increase the acidity of the products. This acidic environment helped inhibit the growth of harmful microorganisms and contributed to food safety. Overall, fermentation improved food preservation, enhanced sensory qualities such as taste and aroma, and extended the shelf life of the products, making it an important process in food production and storage.
Through this simple activity, we learned how microorganisms play an important role in fermentation and food preservation. By observing mango wine, kimchi, and sauerkraut, we saw how microbial activity caused changes in pH, odor, texture, color, and gas production over time. These observations helped us better understand how fermentation works and why it is important in food production and safety. In a world that often focuses on what is visible, fermentation reminds us that some of the most important transformations happen unseen. Through the work of microorganisms, simple ingredients become preserved, enriched, and transformed. And that concludes our presentation. Thank you for joining us in exploring the hidden world of microbiology.